Most people think fasting is about calorie restriction and weight loss. That framing dramatically undersells what the science shows. A landmark review in the New England Journal of Medicine, one of the most prestigious medical journals on earth, documented that intermittent fasting triggers profound changes in metabolic switching, cellular repair through autophagy, inflammatory marker reduction, neuroplasticity enhancement, and cardiovascular risk factor improvement, all independent of weight loss. Some of these effects, particularly the activation of autophagy, cannot be replicated by any known supplement, exercise protocol, or dietary pattern. Fasting is, in the most literal sense, a biological reset that evolution built into our physiology, and the research suggests most of us are never activating it. For the vast majority of human evolutionary history, humans did not eat 3 structured meals per day plus snacks. Food was intermittently available, and the human body developed sophisticated metabolic machinery for functioning, and functioning well, in a fasted state. The metabolic switch from glucose to ketone bodies as a fuel source is not a modern hack. It is an ancient adaptation that produces measurable improvements in cognitive clarity, cellular repair, and stress resilience. Modern constant feeding patterns keep the body in a perpetual anabolic, pro-inflammatory state that may never activate these fasting-induced pathways. The science of fasting beyond weight loss is really the science of switching on systems your body was designed to use. The science of fasting has undergone a revolution in the past decade, driven by landmark research from teams led by Valter Longo at USC and Mark Mattson at the NIH. Today's sources include reviews in Cell Metabolism, Nature Reviews Neuroscience, and the New England Journal of Medicine, representing the highest tier of peer-reviewed scientific publishing. Contemporary eating patterns in industrialized nations have created a situation biologically unprecedented in human history. Most people spend virtually every waking hour in a fed state. 3 meals plus multiple snacks means blood glucose and insulin are chronically elevated. The body never shifts into fat-burning mode, and crucially, the cellular maintenance processes that require a fasted state, autophagy chief among them, are perpetually suppressed. Autophagy is the cellular housekeeping process by which the body degrades and recycles damaged cellular components. Its suppression is implicated in accelerated aging, neurodegenerative disease, metabolic dysfunction, and increased cancer risk. Modern eating patterns may be one of the most significant drivers of chronic disease. The biggest myth about fasting is that it causes muscle loss, that going without food for 16 hours will cause your body to catabolize its own muscle tissue for fuel. This fear has almost no scientific support in the time ranges relevant to intermittent fasting. Muscle protein breakdown does not meaningfully increase during short-term fasts of 16 to 24 hours in healthy adults. The body preferentially mobilizes stored fat, and growth hormone, which is strongly muscle protective, actually spikes during fasting. The muscle loss concern becomes relevant only in prolonged multi-day fasts. For the 16:8 or 5:2 protocols most commonly studied, resistance-trained individuals show no muscle mass reduction compared to continuous caloric restriction. Mark Mattson and Rafael de Cabo at the NEI published a comprehensive review of intermittent fasting research in the New England Journal of Medicine that synthesized decades of both animal and human research. Their analysis documented that intermittent fasting produces improvements in blood pressure, resting heart rate, LDL cholesterol, triglycerides, blood glucose, and insulin resistance, independent of weight loss. In animal models, the same protocols extended lifespan and delayed onset of cancer, diabetes, and neurodegenerative disease. In human studies, inflammatory markers including CRP and Illinois-6 were significantly reduced. The authors concluded that intermittent fasting has therapeutic potential for a wide range of chronic diseases and should be studied as a formal clinical intervention. One of the most significant non-weight benefits of fasting is the activation of autophagy, a process so important that its discoverer, Yoshinori Ohsumi, received the Nobel Prize in Physiology or Medicine in 2016 for characterizing it. Autophagy literally means self-eating. Cells degrade and recycle damaged proteins, dysfunctional organelles, and potential cancer-initiating cells. It is the body's primary cellular quality control mechanism. Animal studies show that autophagy is strongly induced after 12 to 16 hours of fasting, and in human studies, markers of autophagy activity increase significantly in white blood cells after 24 hours without food. No pharmaceutical or dietary supplement has been shown to reliably activate autophagy to the same degree as fasting. The neurological effects of fasting are among the most compelling non-weight benefits documented in the literature. Mark Mattson's review in Nature Reviews Neuroscience documented that intermittent fasting increases BDNF, brain-derived neurotrophic factor, production in the hippocampus, the brain region most critical for learning and memory. In animal models, IF protects neurons against metabolic and oxidative stress, reduces amyloid plaque accumulation, and improves cognitive performance. The mechanism involves the production of beta-hydroxybutyrate, a ketone body, which crosses the blood-brain barrier and serves as a signaling molecule promoting neuroplasticity. These findings have significant implications for the prevention of age-related cognitive decline. The metabolic and cellular benefits of fasting accumulate along a time axis that maps to the duration of the fast. Understanding this progression helps calibrate expectations and choose the right fasting protocol for your goals. The first 12 hours primarily deplete liver glycogen. From hours 12 to 16, fatty acid mobilization intensifies and ketone production begins. From hours 16 to 24, autophagy is robustly activated and growth hormone surges. Beyond 24 hours, the changes deepen further, but for most people and most goals, the 16-hour mark represents the optimal cost-benefit threshold. Comparing health and performance markers between people practicing structured fasting and those eating on a standard 3-meal plus snacks pattern reveals consistent advantages across multiple domains. The data is particularly striking for inflammatory markers and insulin sensitivity, two of the strongest upstream predictors of long-term chronic disease risk. Cognitive performance markers, including reaction time and working memory under fasting conditions, also show improvements relative to post-meal states, consistent with the glucose vs ketone fuel source research. The evidence-based fasting framework presents 3 protocols organized by goal and lifestyle compatibility. The 16:8 protocol, 16 hours fasting, 8-hour eating window, is the most studied and most accessible entry point. It is typically implemented by skipping breakfast and eating between noon and 8 p.m. The 5:2 protocol involves eating normally 5 days per week and restricting to approximately 500 calories on 2 non-consecutive days, well supported for metabolic and longevity markers. 12-hour fasting, simply stopping eating after dinner and not eating until a full 12 hours have passed, is the minimum effective dose for circadian alignment and insulin regulation. The fasting habit loop requires careful architecture because it involves removing a behavior, eating, rather than adding one. The cue is your designated eating window boundary. The routine is the deliberate decision not to eat, and the reward is the combination of accumulated evidence of better energy, focus, and physical composition over time. In the early stages, cognitive reframing is critical. Hunger during the fasting window should be understood as a hormonal fluctuation, not an emergency. Ghrelin, the hunger hormone, follows a rhythmic pattern that responds to your eating schedule. Within 2 weeks, most people report that their fasting window feels natural. Start your fasting habit with these 5 concrete actions. This week, choose a 12-hour window to not eat. For most people, this means finishing dinner by 8 p.m. and not eating until 8 a.m. Track when you actually eat for 3 days using any food journal to establish your current eating window. Push breakfast back by 30 minutes each week until you reach your target window. During fasting hours, drink water, black coffee, or plain tea. These do not break a metabolic fast. And critically, do not start with a 24-hour fast. The evidence strongly supports gradual extension of the fasting window rather than dramatic initial restriction. The highest risk moments for breaking a fast prematurely are social situations, habitual snacking triggers, and late-night eating. Pre-planning your responses to these specific situations using implementation intentions dramatically increases the likelihood of maintaining your fasting protocol, especially in the early weeks before the habit is fully consolidated. Fasting's benefits extend far beyond weight loss. At the cellular level, it activates autophagy, the body's primary quality control process, which no supplement can replicate. At the metabolic level, it improves insulin sensitivity, reduces inflammation, and lowers cardiovascular risk markers independent of caloric restriction. At the neurological level, it increases BDNF production and produces neuroprotective ketone bodies. The 16-hour fasting threshold is where most of these benefits converge, and within 2 weeks, the hunger adaptation makes it sustainable for most healthy adults. Tonight, set a kitchen close time. Finish eating by 8 p.m. and don't eat again until 8 a.m. That's a 12-hour fast, the minimum effective dose. Build from there. If this science was new to you, subscribe to Habits, backed by science. We cover the research that changes how you live. Your cells will thank you.